Start with acid fast staining. Acid fast staining or ZN staining introduced by Paul Ehrlich. It is modified by Steele-Nielsen, hence it is also known as ZN staining or Steele-Nielsen staining. It is mainly used for the demonstration of mycobacterium species. Mainly for mycobacterium species, it can also be used for the demonstration of other bacteria like other organisms like Nocardia, Cryptosporidium, spores, etc. So here we are doing mycobacterium, staining of mycobacterium tuberculosis. You should be careful because we are doing some sample of TB patients, so you should be careful. So here you will get a smear acid fast. So it's a airtight heat fixed smear should be provided. The things you have to bring for the experiment is same, that is a watch, then an eosin. The watch is necessary, then eosin pencil, then a sky blue pencil. This is a light blue pencil, eosin pencil and light blue pencil. Then the normal pencil for pencil and compass for drawing the circle. So acid fast staining. The aim of acid fast staining is, so before that, in acid fast smear it is written as AFB. AFB is written on the underside of the slide. AFB is written on the underside of the slide and the smear will be on the opposite side. Should be careful. AFB for acid fast staining or ZN staining. AFB is written on the underside of the slide and the smear will be on the opposite side. Should be careful about that. So we have to keep the smear on the rack. Don't get confused with the side. You should keep the smear on the rack. So primary staining. Primary staining with the, here we are using carbolfuxin. So primary stain. First I will introduce the stains used. First one carbolfuxin. Steele-Nielsen carbolfuxin. Then the decolorizer used is 20% H2SO4. Decolorizer used is 20% H2SO4 and the counter stain. As it is a differential stain, we have to use a secondary stain or counter stain. That is Loeffler's methylene blue. So keep the smear on the rack. Then first step we are pouring carbolfuxin. That is Steele-Nielsen carbolfuxin. You have to cover the entire slide with the stain solution. You have to pour carbolfuxin and keep it for 5 minutes with intermittent heating. With intermittent heating, that means do not allow the stain to boil or dry. Do not allow the stain to boil or dry. So have to wait for 5 minutes. So primary staining with the carbolfuxin. That is Steele-Nielsen. That is a concentrated carbolfuxin. Steele-Nielsen carbolfuxin. Keep it for 5 minutes. That is intermittent heating should be done. You should be careful. Do not let the stain to boil or dry. So within 5 minutes you have to heat it for minimum 3 times. Only have to done only intermittent heating. Then the gas should be turned off. Then you have to wash with, you have to wait for 5 minutes intermittent heating. Then you have to wash with water. Wash with water. You should be careful. Do not allow the stain to boil or dry. And wash with water. Then next step is decolorize. Decolorize with the 20% H2SO4. You should be careful. You have to use 20% H2SO4. It should be poured over the slide. It should be poured over the slide. Keep it for 3 minutes. You have to wait for 3 minutes. So first step, pour carbolfuxin over the smear. Keep it for 5 minutes with intermittent heating without letting the stain to boil or dry. Then you have to wash it with water. Decolorize with 20% H2SO4. Decolorize with 20% H2SO4. Keep it for 3 minutes. So after primary staining, you can see a pink color in the slide. Because the carbolfuxin have a pink color, so the stain should be, the slide should be in a, or the smear should be in a pink color. So after decolorization for 3 minutes, you have to wash with water and check whether any pink color retains in the smear. You have to check whether any pink color retains in the smear. Again, you have to decolorize with the H2SO4 for another 2 minutes. So decolorization step, first you have to decolorize with the 20% H2SO4 for 3 minutes. After 3 minutes, you have to wash with water. and you have to check whether any pink color retains in the slide. If any pink color retains in the slide, you have to again decolorize with 20% H2SO4 for another 2 minutes. So total 5 minutes. So first step with 3 minutes. If the pink color retains, again you have to decolorize for another 2 minutes. Then wash with water. You have to check the smear. Now it is clear, there is no any pink color. Then we are going to a last step. Last step, that is for Loeffler's methylene. For Loeffler's methylene group over the slide, keep it for 1 minute. Keep it for 1 minute. In this staining procedure, you have to pour the smear over the entire slide. Not like in the gram stain. Gram stain we are pouring only on that smeared area. But here you have to pour over the entire slide. Then again wash with water and keep it for drying in a slanting position. Keep it for drying in a slanting position. So next we can learn about what is acid fastness. This acid fast property is mainly shown by Mycobacterium species. This is due to the presence of mycolic acid on the cell wall. Mycolic acid is you have studied in your theory. Mycolic acid is a wax-like substance present in the cell wall of Mycobacterium species. So this bacteria are difficult to stain. You have to apply heat during staining and you have to use concentrated stains. So with the application of heat and this concentrated stain, they will take the stain. They will not take ordinary stains. They will take the stain, but one stain, they will resist decolorization with the dilute mineral acid. That property is known as acid fastness. So the principle of this acid staining is same. Due to the presence of mycolic acid in the cell wall, this bacteria do not take ordinary stains. So we have to apply heat and we have to use concentrated stains. But one stain, they will resist decolorization with the dilute mineral acids. That property is known as acid fast. It's an important question for you. So primer, once again I will repeat the procedure. Then before that, the aim is to differentiate bacteria into acid fast and non-acid fast. Principle, due to the presence of mycolic acid on the cell wall, they do not take ordinary stains, but one stain they will resist decolorization with the dilute mineral acids. And coming to procedure, primary stain with the Steele-Nielsen's carbofluoxin. It's a concentrated one, Steele-Nielsen's carbofluoxin. Keep it for 5 minutes. With intermittent heating, do not allow the stain to boil or dry. Then, second step, you have to decolorize with the 20% H2SO4 for first step for 3 minutes. Then you have to wash with water and check whether any pink color retains in the slide. Because after primer, the Steele-Nielsen's carbofluoxin is having pink color. So after primary staining, the smear will be having a pink color. So after first set of decolorization, you have to check whether any pink color retains in the smear. So if the pink color retains, again you have to decolorize with 20% H2SO4 for another 2 minutes. Then wash with water. Last step is counter stain with the Loeffler's methylene group. Keep it for 1 minute. Wash with water. Observe under oil immersion objective. So in your observation, you are already studied that acid fast bacteria, they on stain they will resist decolorization. So if acid fast bacteria is present in the smear, it will take the color of the primary stain. That's the color of the primary stain is pink color. So if acid fast bacteria is present in the given smear, it will appear in pink color and the background will be in blue color. If the bacteria is known acid fast, it will take the blue color. So in your observation, you have to write as pink color rod-shaped organisms are present in a blue background. So in observation, we have to write the color and the shape. Pink color rod-shaped organisms against a blue background. In your inference, we have to write it as the given smear contain acid fast bacilli. Observation, pink color rod-shaped organism in a blue background, and in your inference, the given smear contain acid fast bacilli. So you have to study the examples of acid fast organism. Most important examples are Mycobacterium species, that is Mycobacterium tuberculosis, Mycobacterium leprae. Other than Mycobacteria, acid fast structures include Nocardia, then Cryptosporidium, spores, etc. In all this, the methods are same, but the concentration of H2SO4 will vary. In Mycobacterium tuberculosis, we are using 20% H2SO4, but in Mycobacterium leprae, we are using 5% H2SO4. In Nocardia cryptosporidium, we are using 1 to 2% of H2SO4. For bacterial spores, we have to use 0.25% of H2SO4. This is very important. These are your viva-voce questions. The percentage of H2SO4 for TB 20%, for leprae 5%, for Nocardia cryptosporidium 1%, for spores it is 0.25%. So that's about acid-fast stage. Here you can see the dried smear. Here focusing under oil immersion objective, so you have to pour 1 drop of oil. You must be careful, just add 1 drop of oil over the smear and focus under oil immersion object, just 1 drop. Otherwise it is difficult for you to focus. Put on the light. You have to clean the oil immersion objective. Here also we are focusing on oil immersion objective, so you have to clean it. Keep the slide on the stage. Do the adjustments and focus it. First you have to focus the background. It is not easy as gram staining. Gram staining we are getting a culture smear. Here we are taking the direct sputum. We are focusing with the sputum sample of the patient. So first you have to focus the blue background. Then you have to search for the pink color bacilli. First you have to focus for the blue background. Then you have to search for the pink color rod-shaped organisms. This acid-fast bacillus, these are long filamentous bacteria, not like the usual bacilli. These are long filamentous bacteria. The important questions you have to learn from this session. Define acid fast is an important question. Then modifications of Steenius and staining. I think that you have studied in your theory, the Steenius modifications. Then another important, I have already told you that the percentage of H2SO for leprae, for Nocardia, for cryptosporidium and for spores, like that. Here you can see the focused one. Place present is here, it will appear in pink color. So in observation you have to write it as pink color rod-shaped organism acid. Always remember, in observation you have to mention the color, shape and arrangement. In inference you have to report it as gram positive or gram negative, is acid fast or non-acid fast, like that. Here you can see acid fast bacilli. The pink color rod-shaped organism. It is filamentous one, not like the usual bacteria, and you can see the blue background. That is pus cells. So you have to focus the smear like that. Pink color rod-shaped.